Hey guys, welcome back. Today we're going to be starting over. We're going to start over with Chapter 3, which Chapter 3 just essentially, this is Chapter 3. Chapter 3 basically covers over mitosis, meiosis, and essentially the beginning of sex linkage. So most of the time in this chapter, this is mostly something that you've probably all been, you know, covered ever since like Bio 1 days, ever since AP Biology days. So what's gonna happen is we're gonna go over it a little bit more quickly than what we usually do. Uh, but to begin with, we're gonna look at the most we're gonna look at the most common one, which happens in all somatic cells or non-sex cells. And this process, it basically you're generating a identical copy of the cell. So this process generates two daughter cells. Oh, I'm going over that line over there. Generates two daughter cells that are genetically identical to the original cell. So basically, what I have here is two terms that a lot of students sort of mix up during this time. And what I would say, most of the time, people mostly struggle on looking at which one is a homologous chromosome and which one is a sister chromatid. So let's quickly define these. So a homologous chromosome is a chromosome that carries the same copy of the same gene, but from different parental origins. So basically, a homologous chromosome is essentially a chromosome that essentially carries the same exact genes, but it comes from a different parent, and these different parents may have different alleles for each of these genes. So essentially, a homologous chromosome is a chromosome that sort of just has different alleles for the same genes. So we can refer to this as something like this parent from the dad has the big A, and then this from the mom right here, she gave a little a. But obviously you can have the same alleles, but there will be multiple different genes that are being that have multiple different alleles. So you're gonna have different cases like that. And then on a sister chromatid, so a sister chromatid is basically an identical copy of a chromosome. So let's say we're still using this example right here. So a complete copy of a chromosome will just take that chromosome from dad, which is a big A, and it will just copy and paste it in. So then you would have two chromosomes that are both from dad that are big A, big A. So that's basically that. And that's what a lot of students struggle on, defining which one is which. So to begin with meiosis or mitosis, there's essentially the cell has to complete a series of growth phases in order for the cell to properly divide. And this is typically referred to as a cell cycle. So during this cell cycle, there's two main phases called the interphase and the M phase. During this interphase, basically that's where you end up having the growth phase, and essentially G1 phase is normal cell processing phase. And then when you enter S phase, this is typically referred to as the DNA synthesis phase. So basically what happens here is you would start off with a certain amount of DNA. Let's say there's 10 picograms of DNA here. In the S phase section, you're going to end up with 20 picograms of DNA. So then after you leave S phase, you're going to G2 phase, which essentially you're preparing for for cell division by condensing all your chromosomes. So nothing really, you're still maintaining that at 20 picograms of DNA. And then this is when you end up at M phase. During this part, this is when you start splitting off that 20 picograms of DNA into two separate cells that both have 10 picograms of DNA. So by the end of this telophase section, you would have two cells, both at 10 picograms of DNA. Okay. Now, obviously, with this, there's also checkpoints as well at different parts of the cell cycle. These checkpoints are very important to maintain the stability of the cell from it turning into a cancerous cell. So also be aware of that. And typically, if you do not pass a checkpoint or something like that, you're going to end up going into G0, where you're either going to, where the cell's going to undergo programmed cell death, apoptosis, or maybe the cell was supposed to enter G0 and remain specialized or differentiated. So that's basically the cell cycle. And then there are 5 important stages of meiosis, or the M phase. During these phases, what's happening here is that in prophase, your chromosomes condense into these chromosomes, those two sister chromatids, and then the spindle apparatus is starting to form. During the pro-metaphase section, the nuclear envelope disappears, and then this is essentially when the kinetochores and the microtubules start interacting with one another. Then during metaphase, this is essentially where all the homologous chromosomes line up at the middle of the plate. And then anaphase is basically where the sister chromatids are pulled apart to one cell, to one of the poles of the cell. And then telophase is where you reform into 2 different cells with a nuclear envelope reforming as well. But that's basically it. It's nothing too crazy so far for this chapter. And then as we go into the next video where we're going to go over meiosis, that's going to be a pretty simple video too, if you're comfortable with that. So that's it.